Hello, my name is Chen and I am a PhD student at University of Alabama at Birmingham. Today I'm going to present a poster about my project, Gene Fusion Detection and Characterization in Long-Read Cancer Transcriptome with FusionSeeker. So what we did is basically develop this package called FusionSeeker that can be used to identify gene fusions in the long-read RNA sequencing of cancer samples, and also to reconstruct the transcript sequences for those gene fusion events we have identified. We can probably start from the methods here. So this is a figure showing the overall workflow for FusionSeeker, and we take the read alignment file and also the gene annotation file as the input. The first step for FusionSeeker is to identify the gene fusion raw signals, and we go through all the read alignment in the band file, and for each alignment we will do the annotation and see if it is aligned to more than one gene. And if it does, we try to look for patterns in these two cases, if they are on the same strand or if they are on the different strand. And in both cases, we report, it infers the breakpoint locations on gene 1 and then gene 2. And if it does fit gene fusion, then we will record the two gene names and chromosomes and coordinates of the breakpoints and some read length, read mapping quality, some other features. So after we have gone through the entire band file, we will get a long list of the raw signals, and the next step here is to merge them into the candidates. So the merging or clustering process consists of two steps. The first one is to group them based on which two genes are fused together, and the second one is to do the density-based clustering within each gene pair, so that we can separate cluster the raw signals from different breakpoints, and also remove the remaining as outliers. And we also have a filtering process after the clustering that is used to remove the low confidence gene fusion events if they are supported by very few reads or if they have very low mapping quality. And after this step, we will have a list of the gene fusion events, and then for each one of them, we will try to do the third step to generate the transcript sequence by running partial order alignment using all of the reads that are containing sequences from both genes. So after running BS align to do the partial order alignment, we are able to get the consensus sequence, which is very accurate and should have no or very few sequencing errors, and this will be used to present the fused transcript. So eventually the output will be a list of gene fusions and also their transcript sequences. And here on the bottom, we have the benchmark and simulated datasets. Well, we simulated 150 gene fusions, and we run 3 different long-read gene fusion callers to see how many of them can they recapture. And here we benchmark the recall, precision, and advanced score in both ISO-seq and nanopore datasets, and we see that FusionSeeker is able to get the highest advanced score in both datasets. So we have 3 replicates here, and these numbers are kind of the average number from the 3 replicates. And we can also benchmark the transcript sequences. So if we compare to the ground truth, we are able to know the identity of each sequence. And the blue ones are the FusionSeeker's transcript sequence, and they usually have significantly higher accuracy than the raw read itself. And the good thing of having this more accurate consensus sequence is that we can infer more accurate breakpoint locations than what we can do from the raw reads. And here we further benchmarked the breakpoint accuracy for the 3 tools, and we see that on the top for the FusionSeeker's result, we have a very high peak in the middle at 0, which means most of the events are reported with exact breakpoint locations. And remaining, we don't see that peak, which means they have worse breakpoint accuracy than FusionSeeker. And then we move on to the real data sets. So we have 3 cancer cell lines. They have been some previous studies have identified gene fusions using short-read data and validated through PCR. And here we apply 3 different callers to see how many of the previously validated events can be captured here. And we highlighted the highest number among the 3 tools in bold. So we see that FusionSeeker is able to report most number of the previously validated events among the 3 tools, which means it usually has higher sensitivity. And at the bottom, we have the vein plots showing the overlap information from of the 3 gene fusion call sets of the 3 different tools. And some of them can be identified by more than one caller, and some of them are really unique to a specific gene fusion caller. And we try to validate, well, cross-validate in the DNA sequencing data, and we see that the validation rate is highest in the FusionSeeker's unique events, and it is lower here and here. And on the right side, we have the similar analysis done in the MCF7 HiTSeq and also the nanopore datasets, and we still have some in common and also some FusionSeeker specific events. And we designed PCR primers for 10 events that are unreported by FusionSeeker, and we were able to validate 7 out of those 10. And on the right side, we have some features that we really want to highlight for FusionSeeker. The first one is that we can identify the breakpoints even if they are located in the introns. So this is an example for a previously validated event with 2 breakpoints in the same gene. So the first one is located in the exon, which has been reported by all the 3 colors, which is nice. And the second one here, we see a lot of reads also ended here that is located in the intron. This is also validated through PCR, but is only reported by FusionSeeker, but not by the other 2 tools. So that means we are able to report something that cannot be reported by the other method. And another thing is still about the transcript sequences. So similar as what we did here, we can also compare the identity from the FusionSeeker's sequence, then also the sequence from the raw reads, and we do see a higher accuracy in the transcript sequence reported by FusionSeeker. And the accuracy is similar or lower in the JFOS result here and here. And on the right side, we have 2 examples showing the read alignment and also the transcript sequence alignment. And we see a lot of the sequencing errors in the raw reads, but we cannot see any here on the top in the FusionSeeker's result. And similarly, in the nanopore dataset, we also have a lot of the sequencing errors here, but not really in the transcript sequence reported by FusionSeeker. So that means we do have a higher accuracy, especially the base accuracy in the FusionSeeker's result. So that is kind of what I have for FusionSeeker, that we can identify gene fusions with high accuracy using the long-read RNA sequencing data, and we can also report accurate transcript sequences, well, at least more accurate than the read itself. And I really want to thank you for your attention and time.